Hello everyone, I'm Dr. Devyani Day joining you from New Delhi, India. I'm working as a head microbiologist and head infectious disease serology at DR DANGS LAB New Delhi. At the outset, I would like to thank the organizers of the event for inviting me as a speaker in this prestigious event. Now, the ongoing COVID-19 pandemic has turned the world upside down. It's as if we are living in a dystopian society, and amongst the several crises that human race faces today, I will be touching upon the antimicrobial resistance aspect and our constant war against superbugs. Antimicrobial resistance is a growing global problem to which the ongoing COVID-19 pandemic may contribute significantly. Now, with resources deployed away from antimicrobial stewardship, evidence of substantial preemptive antibiotic use in COVID-19 patients and indirectly with deteriorating economic conditions fueling poverty, potentially impacting on levels of resistance, AMR threat remains significant. The high number of patients suffering from coronavirus disease 19 or COVID-19 worldwide admitted to hospitals, wards, and intensive care units are indeed most vulnerable for secondary infections against a backdrop of circulating bacterial strains and of having invasive procedures as putative portals of infection, worsening inflammatory parameters might make it challenging for the clinician to discern COVID-19 severe inflammatory reaction from bacterial secondary infection, setting a low bar in commencing empirical broad-spectrum antibiotic coverage. Though antibiotic prescribing practices might differ between geographic areas, increased work pressure and mental strain on healthcare workers renders it more difficult in adhering to the local hospital antimicrobial stewardship policy in these times. Now, based on a systematic review involving 2,834 patients, Lansbury found out and reported that the co-infection in 7% of hospitalized and 14% of patients admitted to mixed ward or ICU setting. These figures correspond well to findings by Langford et al. Obtained from a living systematic review on bacterial co-infection and secondary in-hospital acquired infection in patients with COVID-19. The findings on community-acquired bacterial co-infection favorably compare to the figures obtained during seasonal and pandemic influenza episodes, for which nasopharyngeal colonizing pathogens such as strep pneumoniae, Streptococcus pyogenes, and Staphylococcus aureus typically cause secondary infections and represent the reason for hospital admission. In contrast, for COVID-19, hospital and ventilator-associated pneumonia seem prominent, with isolates likely to reflect the local institutional ecology and antimicrobial resistance pattern. Further prospective monitoring of co-infections or superinfections will be required to provide a better understanding of such risk and to enable enhanced antimicrobial stewardship. It is noteworthy to indicate that COVID-19 cases that have been administered empiric use of antibiotics may also front on to subsequent complications due to co-infection by antibiotic-resistant bacterial agents. In order to select the appropriate antimicrobials to increase the patient's survival rate and further inhibit the spread of AMR, microbiological examination and antibiotic susceptibility testing of COVID-19 patients presenting with co-infections can be implemented in routine laboratory testing. Moreover, the application of effective narrow-spectrum antibiotics in patients with COVID-19 should be considered wherever possible. Presently, AMR is responsible for approximately 7 lakh deaths every year worldwide as per IACG 2019. Now, the evidence behind these predictions, for instance, during the outbreaks of former emerging pathogen, severe acute respiratory syndrome, that is the SARS epidemic, there was evidence of an increase in prevalence of resistant bacterial infections such as MRSA in the hospital environment. Until now, there are very few data available with respect to cases with COVID-19 contracting bacterial co-infections. It was estimated from separate studies that approximately 1 to 10% of patients with COVID-19 contracted secondary bacterial infections. In another study in Wuhan, 11% of death rate was reported among 99 patients with novel coronavirus pneumonia, and most of the cases, that is 71%, were treated with antibiotics. However, only 1% experienced co-infection with bacteria and 4% had fungal co-infections. Now, what could be the approach to deal this AMR crisis? Either early diagnosis or a suitable treatment option. There is a need for a paradigm shift in ID diagnostics that could significantly reduce the morbidity and mortality, the communicability of the disease, and also tackle the antibiotic resistance apocalypse. Now, there are various factors responsible for increasing the risk of antimicrobial resistance. Firstly, the adequacy of the initial antibiotic chosen for treating the infection. Secondly, de-escalation is not possible before culture report. Now, every culture requires at least a time of 48 hours. So, before that, you cannot narrow down to a narrow spectrum antibiotic, and the patient is given a broad spectrum antibiotic till that time. Thirdly, that is what leads to emergence of resistance. Now, to detect the multi-drug resistant pathogen in a more efficient manner so as to decrease the chances of AMR, we need certain rapid tests which could tell us the organism as well as the susceptibility. To aid in that, we have certain very good state-of-the-art technology that could help us in dealing with such pathogens, namely the bioMérieux film array. This is the only FDA approved multiplex PCR. Now it comes in, it has a sepsis panel and a pneumonia panel. I'll tell you more about the same. The pneumonia panel detects viruses, atypical bacteria, and bacteria found in pneumonia patients, the sample being sputum or bronchoalveolar lavage fluid or endotracheal aspirate. Now you can see the wide array of pathogens detected by one single assay in a specimen. Now coming to the advantages of this assay, now it gives also not only the information on the target pathogens but also important information on antibiotic susceptibility. It tells you about any ESBL or extended spectrum beta lactamase producing organism is present in the sample or not, the carbapenemase producing organism as well as MRSA. Now, the biggest advantage of this assay is that it has a quantitative approach. Now, this quantitative approach allows you to differentiate between the colonizer and the pathogen. One of the biggest causes of antimicrobial resistance is when you give an antibiotic to kill the colonizer. The colonizers are good. They are the flora of that particular anatomical site. So you shouldn't be giving antibiotics to treat, in fact, to eradicate the colonizers. You should be targeting the pathogen. Now, this quantitative approach allows the assay to differentiate between the colonizer and the pathogen. So, it quantifies it. So, it has certain cutoffs. For example, for bronchoalveolar lavage, if the colony count for that pathogen is more than 10 to the power 4, you take it as a pathogen. If it is less, then that is a colonizer and you don't really require to treat it. Similarly, for sputum and endotracheal aspirate and mini-bowel fluid, the cutoff is more than 10 to the power 5 to take it as a pathogen. So to recapitulate, the advantages of this assay is that it targets multiple pathogens, detects co-infections, also interprets the colonizer or the pathogen, and gives you important antibiotic susceptibility information, and all this in a time limit of 1 hour. I repeat, 1 hour. That is path-breaking diagnostics. Now another panel of this film array is the blood culture or the sepsis panel. Now similar to the pneumonia panel, this is also a multiplex PCR and detects multiple pathogens in one go, 27 targets, as well as it tells you about antibiotic resistance in these pathogens detected in a matter of 1 hour. Now, with the help of these state-of-the-art technology, path-breaking diagnostics, it is possible to diagnose and detect multi-drug resistant organisms in a shorter period of time, and that would help you gain a lead time and thus prevent antimicrobial resistance to an extent. Now we all know the pipeline of new antibiotics is drying up, and we are soon about to reach the post-antibiotic era. So here we need to stop, rethink our strategies and boost the power of antibiotics. Now how do we do that? Now we need to think of a viable option where we conserve the power of antibiotics and look for certain alternatives. Now the answer probably lies in the age-old bacteriophage therapy. The 100-year-old therapy is all about bringing in the pre-antibiotic era into the post-antibiotic era. Now, what are bacteriophages? Phages are naturally occurring viruses that attack, infect, and feed on bacteria. They are the most abundant organisms in nature. They are everywhere. They are ubiquitous and found in food, water, air, and even in our bodies. Any environment that has bacteria in it would also have phages that feed on those bacteria. Phage therapy is the use of these phages to treat bacterial infections. An important question crossing your mind right now must be, why have I not heard of phage therapy before? Now, phage therapy has been used for treating infectious conditions for almost 100 years in Eastern European countries like Georgia and Russia. It did not become popular in the rest of the world because after the discovery of penicillin, the global medical industry focused its efforts on the development of antibiotics. The Cold War between Western countries and the erstwhile Soviet Union further widened the gap in world's knowledge of phage therapy. However, as antibiotics are failing in treating an increasing number of infections today, Western interest in reviving the phage therapy as a strong alternative to antibiotics is emerging. Now, how does phage therapy work? Based on the infection, the patient is given a phage preparation that is sensitive to infection. After consuming the phage medication, the phages travel through the body towards the bacteria they are sensitive towards. Once they reach the bacteria at the site of infection, they attach onto the bacterial cells and inject their DNA into the cells. This DNA then uses the bacteria's cell machinery to reproduce very rapidly, making an army of new phages inside the cell. As the number of phages continue to grow rapidly, it is no longer able to contain the invading phages. It bursts, and the new phages spread to other cells of the same bacterium, looking to reproduce, and thus the cycle continues till there are no more bacterial cells left for the phages to invade. Once all bacteria are dead, the phages can no longer replicate and are flushed out from the body. Now, how is phage therapy administered? Depending on the site of your infection, the doctors will decide how the phage medicines are administered to you. It will generally be oral administration or topical administration wherever possible. Now, the current state of using phage therapy in a patient is based on compassionate grounds. Now, as per the Helsinki Declaration, in the treatment of an individual patient where proven interventions do not exist or other known interventions have been ineffective, the physician, after seeking expert advice with informed consent from the patient or a legally authorized representative, may use an unproven intervention if, in the physician's judgment, it offers hope of saving life, re-establishing health, or alleviating suffering. This intervention should subsequently be made the object of research designed to evaluate its safety and efficacy. In all cases, new information must be recorded and, where appropriate, made publicly available. Now, seeing the benefits of phage therapy, the FDA has classified phage therapy, compassionate phage therapy, as an emergency investigational new drug and expanded access. A slice of history, that is George Eliava. Professor George Eliava is the founder of George Eliava Institute, and he, along with Louis Pasteur, started the research on bacteriophages, and bacteriophage susceptibility is being done for almost a century at Georgia, the George Eliava Institute. Now that is George Eliava in the extreme left bottom, along with Louis Pasteur, and up there you can see the team back then, and that's the Eliava Phage Therapy Center. I had the privilege and honor of being trained at George Eliava Institute in February 2020, about which I'll be talking about in the subsequent slides. Now that's me at the George Eliava Institute, where my training was done for bacteriophage susceptibility testing so that we could start the same in India for patients. Scenario of phage therapy in India. Patients wanting to avail phage therapy because of their recalcitrant bacterial infections would either have to go to George Eliava Institute, which is in Georgia, or they would have to send their samples to Georgia. Now, this would require considerable amount of expense and also a lot of time in getting the results. So, as an initiative to combat antimicrobial resistance, our lab first initiated the bacteriophage susceptibility testing in India. Now, this would mean an improved specimen integrity, shorter amount of time, more productivity, increased predictiveness of culture results, and also significantly reduce the TAT, that is the turnaround time, and cost to the patient. Now, as you see, that's an electron microscope through which I am looking and focusing on the bacteriophage on the extreme left as it appears on your screen. Now, the left side, as you see of your screen, is the pharmacy of the Eliava Institute, where you can get the bacteriophage preparations that I've been talking about, and on the right side are the standard bacteriophage preparations. So this is how bacteriophage susceptibility testing appears. This is an in vitro testing in a petri dish, as you see, and lysis means good. Lysis is sensitive, and as you see on the right side, there is certain opacity, that means resistance. So that is how we interpret bacteriophage susceptibility testing. So Dr. Dang's lab is involved in communicating and creating awareness about bacteriophage therapy. We are responsible for sample collection, isolation, and identification of bacterial pathogens along with reporting the antibiotic susceptibility using conventional and automated methods as per CLSI and UCAS guidelines. We provide bacteriophage sensitivity testing of the isolate identified to patients interested in phage therapy in accordance with training provided by Eliava Foundation. Now we use our data pertaining to phage therapy testing and subsequent patient outcomes to further the cause of phage therapy in India. So we understand that there is a current unmet medical need to address AMR crisis. There is absence of approved alternatives. Phage therapy is ideal for compassionate phage therapy use due to its long-standing historical use and publications, solid support by fundamental research, and apparent lack of adverse effects. However, the potential of phage therapy needs to be tapped further for it to emerge as not just a last resort alternative but as a more viable option in treatment of infectious diseases. It is time we go viral with bacteriophages to address the AMR crisis. Thank you.